This module, that is the continuation of the previous module, in which we are talking about batch culture kinetics. So, as I have already told you that in kinetics we will see the relationship of different parameters that gradually change during the fermentation period. So, the major change occur as the definition of the fermentation involve the mass culturing. So, when there is the addition of the inoculum, then there is a gradual change in the number of the cells, or we can say that gradually increase in the biomass of the cell. So in short, we can see when with the respect to the time, as the time will pass, growth will happen. When there will be a growth, then there is the increase in the biomass. So we can say that there is, first there will be a lag phase and then there will be a log phase. So in lag phase, no increase in the number, as I have already told you in previous module, but the size of the cell can enlarge. But especially in exponential phase or a log phase, number of cells also increased. In short, we can say that the exponential phase may be described as the microbial growth pattern. You can see here this simple 2x0. 2 mean double. X0 is the amount of the biomass which we add as in an inoculum. So as there is different multiplication of that, so there are different generations. Mean if one cell become 2 and then in the same period 2 become 4 and 4 become 8. So how these, if one cell and 2 cell is the inoculum level, and then what will be the change? So as increase the generations and there is the same pattern of growth, then there is a exponential change in the biomass with respect to time. So when one cell become 2 and 2 become 4, first generation that become the base of the next generation, and in which the biomass become double to the existing one, which we called as the doubling time, that how much time is required to become the biomass doubled from the existing one. So that is only possible when all the cells autonomously and individually become doubled. So that is only happened when there is a sufficient amount of the substrate and then that have the equal chance of growth. That is only possible when each and individual cells, especially in case of unicellulars, or we can say that in terms of bacteria and the yeast, then the cell become grow, and then how they become doubled. So that is considered as the doubling time.